Hi, my name is Christine Girtain. I'm from New Jersey and I've been working with,
Nathan Diehl, and I'm from Maryland.
and our unit is the science of color and using pH and pigment in photos. We have a list of materials, which we won't read through, that you can read through on your own. And then our standards, lesson objective, and then a little bit about the career connection and the mentor that Nathan has been working with.
Dr. Ravi Jabbour has been working at DevCom over here in Aberdeen Proving Grounds, Maryland, running the CBC lab, which is working on investigating various aspects of cyanide. And he's making a rapid test which detects cyanide uh, in patients for triage. So, in case there's a wildfire and a lot of compounds that are used in the home and other industrial applications, if it gets burned in a wildfire, releases cyanide compounds. And we can develop a test to determine how much exposure they had so that we can help them get aid faster and get medical treatment faster. So that's what the current project that we're working on now, and that's what this unit is based on.
Great. We have a list of some STEM careers as well that list multiple careers from each of the websites there. And then for engagement, the students will explore how nanostructure, color, and magnetic charge are used in nature. Using chromatography, the students will investigate how scientists determine what color pigments contribute to the color we see in nature. And Nathan's going to discuss the flower and bee video a little bit.
So to get students hooked on this idea, we show how bees actually recognize flower colors. Bees see in the ultraviolet, which is not perceptible to human eyes. And so what looks like a pretty flower to us looks vastly different to bees. In fact, the image that you see here is actually yellow flower with a white center, but under UV light, it looks just like a target. So bees are able to target it easier. So we get students excited about and curious about how color plays a role in various aspects of biological life.
The second video that can also be shown is butterflies and birds, talking about how the ability to see blue color happened later in evolution. And so there's not a lot of blue pigment that occurs naturally in nature. Most of the blue that is occurring, like in the blue morpho butterfly, is due to nanostructure and reflecting the life off of that nanostructure and not actual pigment chemically that's doing that refraction or reflection or absorption. There's also some background concept content for the teachers to review through Science Buddies. It gives a great video on paper chromatography and gives some options on how they can also change what they're doing and use different solvents and use different things that they want to extract pigment from. Gives an example of Skittles or M&Ms. In the activity that we chose to do, we're actually having the students extract pigment from leaves, and the students will do that with isopropanol, which is pretty much rubbing alcohol, and they'll be able to do that and then put it on the chromatography paper and look at the separation of the pigments in their individual leaves, which they can either pick out themselves, depending on their age group, or the teacher can supply for them. So they'll get some hands-on background and they'll be able to discuss what pigments were in those actual leaves. The next portion is the explanation portion.
And in the exploration portion, they're going to need their cell phones or pair them up with another student that has a cell phone and download the free app from the either Apple iStore or the Google Play Store called Carolina RGB Colorimeter. This app is basically where you kind of point and shoot at any object, and it will give you the RGB or red, green, blue values for that particular color. In this activity, they're going to use the color key for a pH strip and basically figure out the RGB values for the pH strip and kind of create a standard table. And then they're going to pick unknown samples. And you can do two things. You can either create samples of your own of known pHs and have them experiment with that to see how they can predict the pH from the RGB value of the test strip and compare it to the standards. Or they can collect various rainwater and other water sources from around their areas and make that connection of acid rain and rain in the gutters or a pool or a driveway where they can test the pH of that using their activity. So that's at the middle school level. At the high school level, it goes a little bit further where they're doing the same standardization table of the pH and the RGB app, but they're actually going to then use a spreadsheet program to calculate a line of best fit and an equation of line and use that math equation of line to predict the pH value of the unknown samples. So you would hold off on those rainwater samples until they've made their equation of the line, and then they'll actually try to predict the pH using the RGB value of the pH test strip and see how accurate it is compared to the actual strip. It's just a quick exploration of the app and using pH paper and using it for a particular purpose other than just, pH. They actually get to use technology to make their qualitative of color more quantitative in terms of RGB values.
And then for explanation, students are, Nathan, do you want to go over the explanation?
Sure. In the explanation, they're going to use this knowledge of chromatography and their RGB program to basically, they're going to get a example chromatography paper where it's separated black ink from a pen into its three constituent colors of blue, magenta, and cyan. And they're going to use a graph to basically graph the values based on the distance that they travel. They're going to explain how color is perceived, such like reflection absorption. They're going to kind of quantify their values, and they have a color table. So they have the wavelengths of color where they can predict where red and green would be on that line of best fit. And then they would also get to explain a little bit about the process of separating colors and kind of using the thing, hey, there might be another way to separate colors that's not through chromatography. So it kind of gives you a way for them to explain what they know so far as kind of a formative assessment until they get to another lab and then the final evaluation.
All right, and then for elaborate, we're going to introduce them to gel electrophoresis. If students do not have prior knowledge on how to micropipette, there is a how, sorry, I don't know what this is doing right now. There is a how to micropipette and an introduction on how to set a micropipetter and how to do the micropipetting and some activities the teacher can use for that. In the molecular rainbow lab, the students will actually take various dyes that the teacher has given them, and the gel will be set up so that the wells of the gel are halfway through the gel instead of at one end or the other, so that they're also able to investigate the polarity of the molecules that they're putting into the gel. When the gel runs, usually if it's running DNA as one of the options, DNA is a negative molecule and gets pushed away from the one side of the gel electrophoresis unit and travels to the far side. When we put the wells in the center of the gel, the ones that are positive will travel towards the negative end of the electrophoresis machine, and the ones that are negative will travel towards the positive side of the electrophoresis machine. So they'll go in opposite directions based on their charges. So students will be able to predict charges and see that charge is also one of the ways that molecules can separate within a gel. And then they'll also be able to talk about size of those dyes and be able to see the component parts of those dyes because they will separate just like they did in the chromatography. So it's tying in what they've done previously, taking it to a higher level, and then adding in the component of charge to it. And then for evaluation.
So in the evaluation part, they're going to do a gallery walk. So you can have students create Google Slides and share them where they can present them to the class, or you can do just regular marker, pencil, and chart paper and have them both graph their results from the labs that they did, both of electrophoresis, the chromatography, and the exploration using the Google Sheets, and have them give their information. So in the one they were trying to do their testable question and their hypothesis and their data, and they can present this as a scientific poster presentation. For give feedback from students, they can give them sticky notes and say, hi, I like your data here. I have questions about this, and they can rotate around the room looking at each other's information. If they're doing a Google slide presentation, then they can still put their sticky notes with the person's name and then just stick it on the kid. And so they can read all the feedback, and they can go back and re-edit their slides before submitting it for final grade.
And then these are some of our references that the teachers can use as well.
And there's handouts along with that both covers the lab and the activities.
Yep. And then that's it for us. Okay.